Hello friends, today we will learn about transgenic plants as bioreactors. We will study genetically modified plants can be manipulated to act as bioreactors to produce a wide range of biologically important compounds. These include carbohydrates, lipids, proteins, and the secondary products. The commercial products the commercial products of the plants are useful for industries or for improving the human or animal health. The term molecular farming or metabolic engineering of plants are also used to collectively represent the molecular biological techniques for synthesis of commercial products in plants. Metabolic engineering of transgenic plants Metabolic engineering of transgenic plants is gaining importance in recent years as an attractive alternative to animals and microorganisms for the manufacture of biologically important products. Now let's know about metabolic engineering of carbohydrates. The important carbohydrates are synthesized and stored in different cellular compartments. First point, starch and its derivatives are synthesized in the plastids. Next point, sugars and sugar derivatives are produced in the cytosol and accumulate throughout the cell. Next point, fructans synthesized and stored in vacuoles. Now let's know about the starch. Starch is a polymer of glucose and is composed of amylose and amylopectins. Starch is used as a food, feed, and for industrial purposes. Genetic engineering techniques can be used to manipulate the quantity and quality of starch. Let's know about increasing the production of starch. Biosynthesis of starch in plants is a highly regulated process involving the key enzyme ADP, that is glucose ADP, that is glucose pyrophosphorylase. This enzyme is allosterically controlled by metabolites such as phosphate. ADP glucose pyrophosphorylase in E. coli was mutated to alter its allosteric properties. This mutant gene was transferred and expressed in potato plants. The transgenic potato plants produced high quantities of starch. Now let's know about the synthesis of amylopectin starch. Starch normally contain 20 to 30% amylose and 70 to 80% amylopectin. The relative proportion of amylose and amylopectin determines the physicochemical properties of starch. Amylopectin gels are more stable and therefore are preferred in food processing industries. The enzyme, namely granule bound starch synthase, is responsible for the synthesis of amylose, while starch branching enzyme, that is SBE, produces amylopectin. By using an antisense approach, the enzyme GBSS was inhibited in potato plants. Consequently, amylose free starch, that is starch containing only amylopectin, was produced. Synthesis of high amylose starch. High amylose starch with limited branching is useful as a food, feed, and for certain industrial purposes. It is possible to produce high amylose starch in potatoes by antisense inhibition of starch branching enzyme A and B. SBEA and SBEB. Cyclodextrins. Let's know about cyclodextrins. Cyclodextrins are cone-shaped rings formed by 6 to 8 glycopyranose subunits. They are hydrophilic in nature and can pocket hydrophobic compounds. Due to this property, cyclodextrins are used as therapeutic agents to solubilize hydrophobic pharmaceuticals such as steroids, A key enzyme responsible for the synthesis of cyclodextrins from starch, namely cyclodextrin glycosyl transferase, has been identified. A bacterial gene coding for this enzyme was transferred to potato plants. However, the genetic potato plants did not show any enhancement in the synthesis of cyclodextrins due to various reasons. Now let's know about fructans. Fructans, also referred to as polyfructans, are soluble polymers of fructose. They are synthesized and stored in cellular vacuoles. Short chain fructans are almost as sweet as sucrose. Thus they can be used as sugar subunits in food and soft drinks industries. Another advantage is that fructans are not digested in the gut and therefore they serve as low calorie sweeteners. Transgenic potato plant with fructosyl transferase gene has been developed. Fructosyl transferase being a key enzyme enhanced the synthesis of fructans. In recent years, all oligofructans of sugar beet with suitable or genetic manipulation have been produced. They serve as low calorie sweeteners. Trehalose is a disaccharide produced in plants and microorganisms in response to osmotic stress. Genetic manipulations have been successfully carried out for enhanced synthesis of trehalose to withstand abiotic stress. Trehalose is useful as a food preservative. Some workers are attempting to produce trehalose in bulk quantities for its utility in food industries. Now let's know about metabolic engineering of lipids. Plant oils are useful as foods and for industrial purposes. The industrial applications of plant oils include their use in the manufacture of soaps, detergents, lubricants, and biofuels. There is an involvement of plastid, cytoplasm, and endoplasmic reticulum for the production of oils. After their synthesis, oils are stored in lipid bodies. Acetate from the cytoplasm is taken up by the plastid and converted to acetyl coenzyme A and malonyl coenzyme A. These two molecules undergo a series of reactions catalyzed by the enzyme fatty acid synthase to produce fatty acid carrier proteins with different carbon atoms. Let's see some examples. Laurate is C12, carbon 12. Myristate is carbon 14. Palmitate is carbon 16. Stearate is carbon 18 and oleate is carbon 18 is to 1. By the action of acyl ACP thioesterase, the fatty acids are released and exported to the cytoplasm. Here, they may undergo certain modifications, elongation, desaturation, hydroxylation, etc. These modified fatty acids react with glycerol 3-phosphate to finally form triacyl glycerols. Triacyl glycerol, in short TG, are transported to lipid oil bodies and stored. The oil bodies of seeds also contain proteins called oleosin in the lipid monolayers. And now let's study about production of shorter chain fatty acids. Majority of the plant oils contain more than 16 carbons, example palmitic, stearic, oleic acids. Oils with shorter chains like C carbon 18 to carbon 14 are more useful in many industries for the production of soaps and detergents, cosmetics. It is possible to terminate the hydrolysis of acyl ACP by specific thioesterases and produce high proportions of selected fatty acids. One acyl ACP thioesterase that specifically hydrolyzes Laurel SCP has been isolated from California bay tree, that is Umbe, Umbellularia fornica. The gene encoding this enzyme has been cloned and transferred to oilseed rape. The transgenic plants were found to produce oils with high proportion of lauric acids, that is 12 carbon fatty acids. Now let's know about the production of long chain fatty acids. For the application of industrial oils, glycerols with longer chain fatty acids, that means more than carbon 18, are preferred. Thus, elusic acid, that is 22 carbon containing oils, are useful in industries. However, elusic acid is unsuitable for human consumption. By conventional plant breeding, two distinct crops of oilseed rape have been developed. High erucic acid rape, that is insert here HEAR, and low elusic acid rape, that is LEAR. Clear. Some attempts are being made to genetically manipulate crops to yield oil with high contents of elusic acid. The approaches include to overexpress genes encoding the enzymes elongases and transfer of genes to produce enzymes that can preferentially incorporate erucic acid into triacyl glycerols. Now let's know about production of fatty acids with modified degree of saturations. A fatty acid is said to be saturated if it has no double bonds. Saturated and unsaturated fatty acids occur in nature. Saturated means with on bonds and more double bonds. Unsaturated double bonds with on or more double bonds. It is possible to genetically manipulate the degree of saturation in fatty acid. Now let's know about production of unsaturated fatty acids. Oleic acid, that is C18 carbon 18 is to 1. Rich oils are useful as food, feed, and in some industries. There has been some success in the transfer of antisense gene encoding the enzyme desaturase in oilseed rape and soybean plants. These transgenic plants were found to produce oils with very high proportion of folic acid. Production of saturated fatty acids. Sometimes it is desirable to produce oils with high content of saturated fatty acids. Success has been reported by use of antisense RNA approach. The objective was to reduce the conversion of stearate C18 is to 0 to unsaturated fatty acids such as oleic acid C18 is to 3. The enzyme steroyl ACP desaturase catalyzes the conversion of steroyl ACP to oleic ACP. The transgenic plants developed by this approach were found to contain high contents of saturated fatty acid, namely stearic acid, and low concentration of folic acids. Now let's know about the production of rare fatty acids. There are some rare fatty acids which are industrially important but not for normally synthesized in the plants. Some plants producing the rare fatty acids have been identified and genes transferred for their overproduction. One good example of the production of petroselenic acid that is found in coriander tobacco plants were transferred in with coriander acyl ACP desaturase that lead to a high production of petroselenic acid in tobacco plants. Petroselenic acid is commercially important. On oxidation by ozone, it forms lauric acid and adipic acid. Some success has also been reported in the increased production of several unsaturated fatty acids through genetic manipulation by incorporating genes coding special enzymes, namely front end desaturases. Example: gamma linolenic acid, arachidonic acid, richolenic acid. Biodegradable plastics. Let's know about biodegradable plastics, or in short, bioplastics. Biodegradable plastics are chemically alkanoids, in short PHAS. They are currently being produced in large quantities by microbial fermentation. The structures and the biosynthetic pathways for the production of PHAs now we'll discuss. Among the PHAs, that is polyhydroxybutyrate, PHB is the most important one. Okay. Several experimental studies are in progress to produce bulk quantities of biopesticides in plants. And let's know about the PHB production in cytoplasm. Starting from acetyl coenzyme A, polyhydroxybutyrate is a three-stage pathway involving some enzymes. Let's know about the enzymes. First enzyme is 3-ketothiolase, in short PHA, capital A. Acetoacetyl coenzyme A reductase, PHB synthase. The three genes coding the respective enzymes have been isolated from alkali genes eutrophos and cloned. The cytoplasm of plant cells, example Arabidopsis, contains 3-ketothiolase. Therefore, in the initial experiments, only two genes, PHAB and PHAC, coding acetyl coenzyme A reductase and PHB synthase, were transferred to develop Arabidopsis. By this approach, the quantity of BHP produced was very low. Another limitation was that the plants had stunted growth. Now let's know about PHB production in plastids. The problems encountered in this approach were overcome by transferring all three genes, that is PHA, PHAB and PHAC, of PHB synthesis and targeting them to chloroplast. To achieve this, each gene was separated, fused with a coding sequence of transit peptide bound to N-terminal fragment of Rubisco subunit protein. Rubisco means ribulose 1,5-biphosphate carboxylase. The gene expression was carried out by CAMV 35S promoter. Transgenic Arabidopsis plants with each gene construct were first developed. Then a series of sexual crossing were carried out between the individual transformants. Transgenic plants developed by this approach yielded good quantity of bioplastics, about 14% dry weight of the plant, and in addition there was no observable adverse effect on the growth and fertility of these plants. Now let's know about the production of bioplastics in cotton fibers. Cotton fibers contains the enzyme beta-ketothiolase. Therefore, the genes for the other two enzymes of PHB pathways, that is PHB and PHC, from alkali genes eutrophos, were transferred into meristems of cotton plants by particle bombardment. Adequate sequences of PHB was reported in the fiber of transgenic cotton plants. Now let's know about the production of polyhydroxyalkanoate copolymers. The most important bioplastic of commercial importance is polyhydroxybutyrate. The genetic engineering approaches for its manufacture in plants we discussed. But the disadvantage with PHB production is that it is found as stiff and brittle plastics, since it forms highly crystalline polymers. The other bioplastic composed of polyhydroxyalkanoate copolymer is a polymer made up of longer monomers. It is less It is less crystalline and more flexible compared to PHB. PHA are produced from the intermediates of beta oxidation of fatty acids, notably 3-hydroxyl coenzyme A. Some success has been reported in the production of PHA through genetic manipulations of peroxisomes and glyoxisomes. Now, let's know about the genetically engineered plants as protein factories. Molecular farming or metabolic engineering of proteins in plants is a novel approach that makes plants to serve as living factors for the production of proteins. Use of animals and animal and microbial cell cultures for the bioproduction of proteins. Now, we'll discuss. Production of human or animal proteins by plants is more recent and novel approach. The transgenic plant have some advantages as bioreactors. First point is low cost of production. Second is eukaryotic protein processing. Third is unlimited supply. Fourth is no spread of animal borne diseases. Fifth point is reduced time to market. Sixth is safe and environmental friendly. Seventh is no need for skilled workers. Now, genetically engineered plants can manufacture a wide range of proteins. These include enzymes for industrial and agricultural purposes, lysosomal enzymes, antibodies, subunit vaccines, other biopharmaceuticals or medically related proteins. The general approaches for the production of desired proteins in plants are we first discussed. Okay. Now let's know about the approaches for protein production in plants. In general, methodology for the production of proteins in plants involves some aspects. First is selection of crop species. Second is choose of tissue. Third is expression strategies. Fourth is post translational translations processing. Fifth is recovery strategies. Now let's know about the selection of crop species. The most important consideration for choosing a plant as a bioreactor is that it should be readily manipulated to produce a stable transgenic line with stable expression. It can be stable expression. Tobacco, the most preferred plant as a transgenic bioreactor. Let's know about the plant tobacco. Tobacco is the most widely used plant for the bioproduction of proteins by researchers as well as biotech companies for some reasons. Let's know about the reasons. It can be easily engineered and transformed. Second point is tobacco is an excellent biomass producer and about 40 tons of fresh leaf or leaf per acre land against 4 tons by rice. Seed production is third point is seed production is very high, about 1 million seeds per plant. Fourth point is tobacco can be harvested several times in a year. The major limitations of tobacco plant are it is a regional crop and relatively labor intensive, and it is not suitable to be fed to humans or animals. Other crop species are bioreactors. For the development of edible products, example vaccines, plants such as potato, tomato, banana, maize and lettuce are used. Some workers prefer to produce proteins in the seeds of plants such as canola, soybean, corn, rice and barley. This is mainly because seeds can accumulate and store good quantities of proteins. Now let's know about the choice of tissue. The preferred tissues for protein production by plants include leaves, storage organs, and seeds. This choice of tissues mainly depends on some factors. Let's know about these factors. First is compatibility with the desired protein. Second is correct processing. Third is stable accumulation. Fourth is efficiency of recovery. Many proteins have been produced in leaf, Example lysosomal enzymes, mammalian antibodies, human serum albumin. In recent years, seeds have become an attractive high protein production example vehicle due to high storage capacity and stability. It is no surprise that some of the seed proteins retain their biological activity for several years. One limitation of seeds is that it is sometimes difficult to recover the protein. Now let's know about the expression strategies. There are two major approaches for the transgene expression in plants to produce protein stable integration approach and use of plant viruses as transient vectors. Now let's know about the stable integration approaches. This strategy is most suitable for the bulk production of proteins, particularly in leaves. In this approach, the transgene is regulated by a strong constitutive promoter such as 35S promoter. Another alternative to use tissue specific promoter by restricting the transgene expression to a particular tissue, example seed, the yield of the protein can be substantially increased. In addition, the protein is stable for a long time from weeks to years. Second point Second point is transient expression by using plant viruses. Bioproduction strategies involving viruses are useful for the protein production at the desired period. By genetic manipulations, it is possible to insert the desired gene into the code protein gene of the virus genome. The so developed infectious RNA viruses are introduced into the plants for the production of proteins. Transient expression system by viruses has been successfully used in tobacco plants by employing tobacco mosaic virus. Tobacco plants at an appropriate age were inoculated with genetically modified, that is tobacco mosaic virus TMV. Recombinant protein could be extracted within 2 to 3 weeks of harvesting. Now let's know about post translational processing. The ultimate purpose of the use of plants as bioreactors is to produce proteins with native conformation, good biological activity, and biocompatibility. The specific folding of protein with its disulfide bonds and suitably modified amino acids is important for this purpose. The changes that occur after the protein is synthesized are collectively referred to as post-translational modifications, example phosphorylation, hydroxylation, carboxylation, glycosylation, proteolytic processing, etc. Variations in post-translational changes are often limiting the successful production and use of recombinant proteins either in plants or even in other systems. The major limitations of plants with regard to post-translational modifications of proteins are listed. Glycosylation is different in plants. Plant glycosylation results in the production of complex glycans containing fucose or xylose, which do not occur in humans. Plants cannot perform specialized carboxylation necessary for certain clotting factors 2, 7, 9, and 10 and enzymes, that is protein C. Due to these limitations, many times protein of transgenic plants are inefficient or less efficient in their function, besides exhibiting immunogenicity. In recent years, cloning of genes responsible for post-translational modifications of proteins, example, glycation, carboxylation, have met with success to produce more biologically active proteins. Now let's know about recovery strategies, that is purification strategy. A rational approach for a cost-effective purification A rational approach for a cost-effective purification system is desirable for the recovery of the protein. produced in transgenic plants. The degree of purity is dependent on the purpose for which the protein is produced. For instance, industrial enzymes in general do not require high degree of purity. Sometimes purification may not be necessary at all. A good example is the production of enzyme phytase in seeds. Phytase acts as phytate and increases the ability of phosphate to animals. For this purpose, milled transgenic seeds containing phytase can be directly added to the feed. This totally avoids the purification processing cost. For a great majority of proteins, particularly pharmaceutical proteins, purification strategy is absolutely required. It is therefore necessary to develop efficient methods for downstream processing. Now let's know about the affinity tag based purification. The use of affinity tags for purification of recombinant proteins has been described elsewhere, and a similar strategy works in plant systems as well. The technique involves the fusion of desired protein with another protein or peptide which can bind to ligand, and thus the protein can be isolated. Following purification of the desired protein, the affinity tag can be removed. Some workers have been successful in purifying the enzyme glucocerebrosidase by this approach. Now let's know about the purification through compartmentalization. It is possible to direct the accumulation of proteins in a specific subcellular organelles. This is achieved by using signal peptides or fusion peptides. Once the protein is localized, the technique of subcellular fractionation can be used for preliminary isolation of protein followed by its purification. The human neuropeptide leukoequiphalin has been compartmentalized in the endoplasmic reticulum and vacuoles by this strategy. Then by appropriate purification technique, it is isolated. Now let's know about the protein purification by production in oil bodies. Oil bodies are natural subcellular organelles that store triacyl glycerols. Oil bodies are composed of triacylglycerols enveloped by a phospholipid monolayer. A group of proteins referred to as oleosins are associated with the surface of the oil bodies. Oil bodies are found in the oil or seeds that represent the storage form of energy as TG. Oil seeds contain oleosin at a concentration in the range of 2 to 10% of the total seed proteins. Oil bodies can be used for the production of foreign proteins in plants, the development of transgenic plants, and the purification of the desired protein by employing oil bodies we will discuss. A transgene with oleosin and desired